There we go. Okay. All right. Good morning. Um, I had a bit of a scare this morning because I didn't realize it, but I had not quite tightened my bottle enough before packing my laptop and everything in and sticking it in there and leaving it in the car. So, you ever put on a wet sock? Kind of feels like what I'm doing with my hand right now because this is soggy. So thankfully, it looks like all my electronics are still working, and there was enough padding and everything. And my bag's a little damp, but not. I don't think anything's ruined. So I'm very thankful for that. Okay, so we'll talk more about water and disinfection in a moment, but I would like to get a little bit of feedback from the exam. Already had one question, curious about that first problem with the iron hydrolysis. How did you approach that? We will talk through each of the problems. I like to do that to once I hand back the exams as a way to really connect with you, clear up any issues on the material, and make sure we're on the same page. So we will do that once I get the exams graded. I'm going to give it a try to have them done by the end of fall break. Not going to promise that, but that'll be what we'll try to do. I do give partial credit. What I'm usually doing is looking for the process, right? Did you follow the coherent process? And I'll explain a little more when I hand back the exams, but typically you can expect something like, if you've got one major concept wrong, but everything else looked correct, and you had a good process otherwise, that might be 20 to 30% off, so you'd get 70 to 80% of the credit. If it's just like one little calculation error, you flipped a negative sign or something, then it would be like 1 point off on a problem, kind of a thing. If you've just got repeated mistakes and you missed all of the concepts in the problem, but you gave it a really good effort, and I could tell you were spending a lot of brain power, you still probably get 25% of the credit for making the effort and showing me what you know, even if you're in the wrong direction. So you can expect partial credit on anything you gave a good faith attempt for. But otherwise, I do want to hear a little bit more how you felt about it. So I've got a Kahoot to sort of survey to play. So log in with your 8-9 numbers for this. And this will just kind of be how you felt about it, how you felt about your preparation, stuff like that. And if you've got some other questions about the exam itself for now, happy to chat with you about it, even though we'll have a deeper dive later. Looks like that should be everybody. Okay, there's no real right answers or wrong answers, so don't worry about that. It'll suggest a right answer or whatever. But so how did you feel about the length? Is it too long but manageable, just right, or pretty short? Okay, pretty good. I like to see that a lot of people thought it was just right. Never a good thing if people feel like it's too long. Understand sometimes it just happens. But okay, how'd you feel about your preparation for it? Okay. Well, you know, the blue is the worst, and I'm glad that's not the case here. So I always feel like it's the worst thing when you feel like you're ready for it, and then it just didn't seem to help, right? Great. Okay, how about the difficulty? I kind of personally reflecting on it felt like it might have been on the little bit more challenging side, but well, that's good. Glad some of you felt like you really had it mastered, and most of you felt it was manageable even if challenging. So that looks great to me. And again, this yellow is sort of the worst, right? So you walk out the door and you're like, oh. Okay, in terms of the topics, how do you feel about mass balance reactor type kind of stuff? I'll just have a couple more, you know, each category here that we sort of tested on, and then get a bit of feedback. Okay, one feeling like you have no idea, so let's, you know, please do feel free to reach out to me, set up some office hours, or come to the office hours, either one. Be happy to get that cleared up. Okay, how comfortable about chemistry kind of side of stuff? All right. And just by the way, that first problem, you didn't have to do any chemistry. It made it think you might, but you didn't need to know anything about the iron hydrolysis. So if you're feeling like you had no idea about that, you're fine. As long as you did the mass balance side of it, we'd be just fine. Sedimentation. I think there was just only a little bit there, but you know, concept-wise, you feel okay about that? Okay. Pretty good. And coagulation, flocculation. So this would have been tied. I think the problem had this tied together with the sedimentation, but there's kind of two parts to it. And this one really gets more to the mass balance side of things too. All right. And I think last, well, okay, the two filtrations. So we've got granular and then membrane. A lot of questions today. Okay, maybe the more recent the topic, the better, huh? Well, granular filtration is also pretty straightforward. So. All right, membrane. Okay. And I could just take these and grade them accordingly, right? So the more confident, the better the grade. No. All right, so we've got some winners or whatever they say, but okay. Appreciate that. Thank you for the feedback. And happy to hear more feedback. If you've got any complaints you want to voice right now, or when we go over the solutions, happy to hear them and talk with you about them. Okay, so today we're going to start our section on it's more like the chemical treatment in some sense. So the first phase of this class is more like physical treatment technologies, plus some background, just a refresher on mass balances, things like that. And this section will be more like chemical treatment processes. We'll do disinfection, we'll do water softening. If I get ahead of myself, maybe we'll include a little bit of adsorption stuff, but scheduled we just have the disinfection and water softening. Then the last part of the class will be more biological. So wastewater treatment is largely biological, but plus some of the physical, plus we usually disinfect. So wastewater sort of wraps a lot of things into one. It's just adding the biological type steps at that point. So when we think of disinfected water, first question I like to ask is, what's the difference between disinfected and sterilized? So would you guess that my leaky water bottle, presuming it didn't leak, and let's assume that I had cleaned it before filling it with water, and let's say I filled it from one of the water spigots on, in PFT or something. Would you say this is sterile? Some no, some maybe yes, I'm not sure. Grimacing and sterile, what is that? Anyone think it's sterile? Like for sure? Okay, good. Disinfected? And some non-answers, just like, I don't know, I don't trust that bottle over there. Okay, so do you think this should be, what's coming out of the water tap should be sterile? Okay, some yes. How many think it should be sterile? Like by design? Okay. All right, how about disinfected by design? So hopefully we all think it should be disinfected coming out of the tap. That's what we'd hope, right? Okay, so the difference here is that sterile, when we sterilize something, we're really talking about all organisms in there, and we're talking about either killing them or causing them to be unable to reproduce. So we sterilize our pets pretty often, cats and dogs. We sterilize them so that they're not accidentally making a whole bunch of new kittens or puppies that we actually don't want to care for. We'd rather, that not happen everywhere with every single pet, right? We'd have a lot of pets. So that's one aspect. So if we take ultraviolet light, for example, and blast some bacteria with it, chances are not all of them will die directly at the doses we give. Rather, they're just going to have damaged DNA and not have the capacity to reproduce and make more bacteria. So they might live another few hours or maybe a day or something, but they're not going to be able to cause an infection. So if we do that to all of them, we would sterilize it. Now, disinfection is more about doing that to organisms that could cause an infection to the point where there's not enough of them left to cause an infection. So this water here, I can promise you, even if it did just come out of the tap, is full of bacteria and viruses. And I'm drinking it, right? So if you were to take bottled water or water from the tap, there's a very, very good chance there's lots of living organisms, microorganisms, especially viruses. There's just so many of them around. Now, these viruses are probably, most likely, the type that might infect algae or some other thing, right? So they're very, in the water pipes, there's lots of organisms growing on the pipes that have been able to withstand the bit of the residual chlorine that's there. The deal is, it's disinfected to the point where there's no longer enough infectious organisms that are capable of reproducing that could harm me. When we sterilize water, maybe we put it in a pressure cooker, something like that, we blast it to the point where we are confident that nothing ever is still alive there. That's what we do in the lab if we're trying to do some sort of bacteria test. We need to sterilize everything so that when we try to grow something, we know that the only things that are going to grow is the stuff that we were putting in there. We don't want random contamination occurring. So we sterilize a lot of stuff in my lab. Disinfection is sort of my specialty, so we're gonna have a little fun with this topic. So disinfected, then, it would actually be okay if you told me there was 100 Vibrio cholera cells in this bottle, and you could tell me with absolute certainty that there was exactly that many. It's a pathogenic organism. If you were able to tell me 100% confidence that there's exactly that many in here, I would enjoy drinking the rest of this just because it would be funny. And I could do that, and you'll see why in a minute, but because that's actually not enough to get me infected. There's no way that number could cause an infection based on what we know about Vibrio cholera. All right, so that's some interesting things to think about that, because the disinfection then is at infectious levels. Oops, what did I do? Okay, I think I just didn't click. Okay. So just a quick reorientation with where we are in water treatment, typical water treatment plants. This is, again, a typical surface water treatment. We've covered the sedimentation. We've covered the flocculation, coagulation, sedimentation again. Then we'd have some filtration typically. This is sort of a generic design. And here we are at disinfection. So at this point, we've intentionally removed a lot of the contamination, a lot of the particles that organisms could hide behind or be trapped within. And so we have pretty clean water, but we've not necessarily taken care of any of the viruses, and there's probably a good potential for bacteria also to be floating around. So here's where we would add a disinfectant, and you see it's drawn as a plug flow reactor. That is quite typical for disinfection processes, and if you stop and think about that, we'll come back to that concept, but that makes a lot of sense when we want a high percentage removal. So we want 99.9% or whatever as sort of the safety to make sure that there's no more bacteria or viruses there at infectious levels. We will often then add fluoride and send it through the disinfection system. Any other polishing steps, we will talk about water softening, will also happen in sort of this area. So softening would likely come before, probably before the disinfection. We'll cover it next. I guess maybe we're a little out of order in that sense, but it would be in the same general area. And then we might also adjust for corrosion control, adding a bit of orthophosphate or something. Okay, so back to disinfection, or so on with disinfection. So there's three different types of organisms we really care about, or classifications. First would be viruses, pretty familiar with those these days. There are a wide variety of waterborne diseases that are viral infections, so norovirus, rotavirus, poliovirus. Again, this is the fecal oral path route of transmission, so you can get norovirus if you're drinking from a contaminated water supply. You could also get it if you had somebody serving you Chipotle and their hygiene was not so great and they had norovirus at the time, right? So it could be transmitted that way. It's probably the more often you hear it in that sense, but certainly could also be from the water. And a lot of these really don't take too many viruses. Maybe, I think it's on the order of like somewhere between 5 to 50 viruses for an infection for norovirus. Something like that. Then we have bacteria. And unlike the viruses, bacteria are actual living organisms that are capable of reproducing, multiplying. Usually a bacteria will need some sort of specific environment. A lot of the types that can infect us are infectious precisely because they can generate a lot of themselves. They can reproduce rapidly in a particular condition. So if you've heard of like the flesh-eating bacteria or whatever, that just means you've found a bacteria that can get into your skin or under the skin or something and multiply there. A lot of bacteria won't be able to do that. We are colonized with bacteria of all types, lots of types, that are capable of living on the surface but not consuming our flesh, right? Not actively using that as its food, not getting underneath and growing there. Our bodies are protected generally from that. If you get bacteria in your eye, there are certain types that could start propagating there and give you pink eye. It's an infection. Technically, that's usually some sort of fecal matter. So if you've gone swimming in a lake or something and there's a lot of bacteria there and you got some in and started growing, or if there was a hygiene mishap with somewhere along the line, something like that, you'd have that would be a source of infection potentially. So for getting your stomach infected or getting giving you a food or waterborne illness, you actually the bacteria will have to withstand going through the stomach and the acidity there, and then causing an infection somewhere later, typically. And that would be true of the viruses as well. So viruses, they don't have much protection. They are, just kind of like proteins, basically. So if they can withstand the stomach acid, they're probably going to pass through and be okay. So a fair amount of them will be, or have potential to do so. The bacteria, they're a bit different, right? They have a cell membrane. Their physiology is a bit different. Some of them are going to be pretty vulnerable to acid, some a little stronger. A few of them could even make little cysts. Now, bacteria, we can make a distinction between a food infection, which is what most of us are actually talking about if we say, oh, we got food poisoning, and actual food poisoning, which is there was something toxic that's acting as a poison that gives a pretty immediate poison reaction, right? So if you are poisoned, you become ill because there's some chemical that's poisoning you. It's toxic. You generally have an almost immediate response. So if you get a food infection is really the type of thing that I'm talking about here, but you can also get it from water, which is why we're talking about it. So we'll say food, or technically water, infection. That's usually what people are talking about when they say they got food poisoning. Whereas food poisoning is what happens when, let's say, botulinum bacteria produce botulinum toxin, or and then you get botulism. That it's a poison. It causes paralysis and bad stuff. So if you were to consume that, you'd immediately be poisoned by the stuff that the bacteria produced. A food infection or waterborne infection would be you've ingested the bacteria and they're causing you to be sick, and that could mean they're making that toxin, right? Actually, there's a reason we're not supposed to let infants have honey. It's because the botulinum bacteria is actually fairly common in honey, naturally occurring, and it just is so vulnerable to stomach acid that anybody with a mature stomach won't have any issue. It'll negate the ability for the botulinum bacteria to live or produce anything, and our bodies handle it no problem. If in an infant it passes through the stomach and then starts growing, later on in the intestines or something, then it can start producing the toxin, and that would be typically fatal for the infant. So a lot of this has to do with our stomach as a protective agent, and also has something to do with how likely is a given bacteria going to be able to multiply and have enough of them to do so. So I mentioned earlier, Vibrio cholera happens to be very susceptible to low pH. So we'll say this is very acid susceptible. And so it would require anywhere something like 10 to the 5th to 10 to the 6th or more cells to cause an infection. So it really requires, and I might even have that wrong, it might be 10 to the 6th to 10 to the 7th, on the scale of 1 million Vibrio cholera cells to give somebody an infection. So when I told you if there was 100 of them in here, I'd not be worried about drinking it, because I know I have stomach acidity that's going to kill so many of them. Even if one survived, that's probably not enough to reproduce in time to actually give me a real infection. My body's defenses would handle it. E. coli, some of these can produce a toxin. Some you can actually have that infection that produces a toxin. That's the bad cases of Salmonella and E. coli, but some of those strains will be pathogenic in the sense, well, they will disrupt you even if they're not producing that toxin. So these can go one way or the other. And these are generally something like 1,000 to 10,000, if I recall correctly, cells required. So these will vary. They'll be different from bacteria to bacteria. Protozoa is another one. These guys are, instead of the prokaryotes, meaning they have a cell membrane, these guys are eukaryotes, meaning they have a cell wall, right? They are slightly closer to animals in that sense. But they are single-celled organisms that act a lot like bacteria. They're just a little bit slower to reproduce. They tend to have a little bit more protection from the environment. So they're usually smaller quantities, a little bit larger in size. So amoeba, for example, might go and prey on bacteria and eat a lot of bacteria. But otherwise, they're still living that single-cell life, right? So these can cause infections. Cryptosporidium, we have that paper. I also posted that quiz now. You'll see it's, I think, due a week from Friday. And Giardia is very similar to Cryptosporidium. These generally cause like a few months of diarrhea for if you get an infection for most people. They can be fatal for immunocompromised people. So in that outbreak paper, you'll see they're talking about its coincidence with the AIDS epidemic was a pretty big problem for people that were actually very vulnerable to that infection if they had an active HIV infection. So these guys really only take something like anywhere between 1 to 5 cells to give you an infection. So it might be as small as few as just one of these. People, you might also have heard of these as hiker's diarrhea. So if you ever go hiking and you drink some water that maybe you didn't clean as well as you should have, or didn't quite boil it or whatever, that would be a common thing because these are just everywhere in the environment. Almost every warm-blooded animal is going to have it in its gut. So it'll be in, these guys will be in most places like that. These are also going to be a little larger, so physically easier to separate. Tend to be a little harder to disinfect. So they can form these cysts and be more resistant to chlorination, for example. So each of these pathogens, each of these types, the point here is they're going to have different vulnerabilities to different conditions. They're going to be infectious at different levels. And so that's pretty important when we go and think about how are we going to design a disinfection process. And before we talk about that, how do we know this? How do we know how many bacteria it took to get somebody infected? What do you think? How would we know that? So it turns out that the CDC would offer young healthy people, maybe college students, like, hey, why don't you come hang out over here? We'll give you $1,000 and a free hotel with Wi-Fi sort of situation. It'll be kind of like a hospital hotel sort of situation. We're going to give you exactly the right food for the week. We'll control that, make sure it's nice and clean, and we need you to take this pill. It may or may not contain some virus, and if it does, it might have a lot of them or a few of them, and if you get lucky, it doesn't have it. It's placebo, or if you get lucky, it's you're asymptomatic or don't have enough of them to give you an infection. But we're just going to monitor your health. We're going to keep you happy and healthy, and you might get sick for a week. And just, bribe people to sign up for an experiment. And it kind of makes me think like, ooh, would I have done that? I don't know. How many do you think you would have faced the risk of getting sick for $1,000? I mean, as long as... There you go, right? So It might depend on when's the last time you've had a food infection, right? It's like how bad it, how willing to face that are you, right? So pretty entertaining the way they have gone about gathering this information. So at the end of the day, we can say that phrase, "the dose makes the poison," comes pretty true here. And to truly disinfect water, what we need to do is reduce these to sub-infectious levels, right? And that's the goal. And then we want to make sure that they stay that way. So we typically will disinfect with some sort of strategy and then add a residual disinfectant. Typically that's chloramine, a little bit of free chlorine, plus a little bit of combined, we'll talk more about it, but chlorine plus ammonia, we'll get chloramines that they react a little slower, they stay in the water a little longer, in order to make sure that there's no way that they can repair themselves and live again. So that's kind of the deal. And our typical strategy then is, it's really hard to discover just one type and then quantify just one type. So typically when we're dealing with detecting them in like a water treatment facility, we don't have really super skilled microbiologists that are capable of propagating each and every one of these pathogens. There's nowhere near that. So instead, what we say is, well, let's use some marker virus. We've got some in my lab that's just a bacteriophage. It's a virus that infects bacteria, E. coli actually. And E. coli is easy to grow. And then if you infect the bacteria with the virus, you can count pretty easily, okay, how many viruses did we have in this amount of water? And we'll use that as a test method. And the water treatment plant won't even do that. What they'll do is say, based on what I've done in my lab, we know that, let's say, UV light kills this virus at some rate. So then they'll design their system. Scientists, researchers have discovered the vulnerability of these different organisms to these different disinfectant technologies. And then all that to say, we'll say, all right, we need to make sure that we have 99.99% removal of viruses, or of this particular virus. And if we design it that way, given this technology, it should apply effectively to all the pathogens. And so we kind of have a technology-based standard in that manner. So as long as we meet this criteria for this particular difficult one, we should be pretty assured that it'll apply to all of them. Now, the good thing is, some of these, like the cryptosporidium and stuff, tend not to accumulate in large numbers. So some of them, if you're getting 99% of them reduced, that's like there's only one left in every cubic meter of water or something. Sometimes even lower than that, and we'll just never be able to detect them. Okay, so this brings us to a bit of history. There was a famous doctor by the name of John Snow. Happened to not have direct relation, and no, I'm not talking about the Game of Thrones guy that doesn't know anything. I do have a brother named John, though, so he knows nothing. Okay, so this is John Snow with the H. He's known as the father of epidemiology, so to speak, and that's for his role in really identifying that this particular cholera outbreak in London in the 1850s was associated with a water transmittance. So it was really the first time anybody had tracked disease in this manner. Epidemiology is the tracking of disease, learning about who's at risk and why, based on tracking the rates of infection or death or some outcome, and usually mapping that. So he made this what was called the ghost map. There's actually a really nice book. It's a historical fiction of sorts. It follows very closely the story, but they add just enough fictional dialogue, like conversation, so that it tells as a story instead of like a readout of facts. So it makes it for a very interesting read, kind of believable, context filled in. But we have a lot of information on this whole thing, so it's very closely mapped to reality of what happened. So I definitely recommend that. I think I have a picture of that in a moment. It's a good book for anybody in the field of kind of public health, disinfection, disease tracking kind of stuff. So this is one of the first confirmations of the germ theory of disease. He actually took a look under the microscope at water because he had some suspicion once he was tracking and making a map of things. And he thought he saw some little organisms swimming around. Turns out he probably didn't have a microscope powerful enough to really see Vibrio cholera. It's the one I was talking about, right? So maybe he saw some algae or something, but whatever the case, he thought that was the case. He was technically correct, and he followed that direction and ultimately was able to stop this outbreak. So what happened was, this particular case in London. So here's this book, and then a map from the book. The map here shown is a real map from what London looked like in the 1850s. This little sector of it, and essentially, if you take a look at these black bars here, each little segment was one person who died at that home. So that's why this is called the ghost map. So is mapping the ghosts or the people that had died. And the scary thing about cholera, Vibrio cholera, essentially what it does, it's not so scary today if we have access to treatment, but essentially our intestines are designed to pump water from whatever food, whatever we're digesting, whatever we've drank, pumping it from our stomach or our intestines into our bloodstream. Like they have cells that are designed to do that and are doing that on some continual basis. So it's sort of dewatering our sludge, to use a water treatment analogy. And Vibrio cholera causes, the infection. The cholera bacteria end up reversing that. They emit some sort of chemical or infect the lining in such a way that reverses that. So then it's taking all the water that's in our blood, dumping it into the intestines. And if you've heard anything about cholera before from stories or movies or anything, essentially the treatment or the disease goes in such a way where you have extreme diarrhea, right? It's essentially dehydrating you and emptying it out your intestines. So there's a thing called a cholera cot, which is basically a cot with a hole in it, so you can put a bucket under the person, because it's really that bad, and you're just discharging so much volume of liquids. And nowadays, if you have access to proper hydration, it's almost never a real issue beyond a lot of discomfort. So if you have an IV, if you have rehydration salts and some water to drink, you're almost guaranteed to be totally fine within about a week, and perfectly restored, no long-term impacts that I'm aware of. I mean, maybe some scarring from the from the ordeal, but just mentally. But back then, they didn't know that, and dehydration kills you really quickly. So if you have the situation, you could have the scenario where an individual and perhaps a whole family gets sick, and dehydration at that extreme degree can kill within hours. So today, everybody's happy and healthy, but you're scared because your neighbors are dying, and tomorrow your whole family died and somehow you were asymptomatic or something, right? That's truly terrifying to think about, and to not know what to do about it. It's the plague, right? It's people were thinking it was the miasma, just the gross feeling smells of sewage in the streets and stuff that was a reality. Turns out that at the Broad Street, just above the Broad Street pump, and here's a three-dimensional assessment of this ghost map. You can see it a little clearer. Essentially, and you'll see that the map itself is still marked. You see this big black line there, but what they've done is they've added a vertical red bar there to kind of highlight what that looks like, sort of quasi three-dimensionally. So they set up this map, and then So we see the utter devastation here in this community. The blue flags are different pumps. So those are pumps for a groundwater wells, probably 20, 30 feet deep, just pumping up by hand. You can get some groundwater to drink from. And John Snow recognized, once he mapped all this out like that, that it seemed very likely that this pump, the Broad Street well, was the problem. And it turns out he was right. So what he did was he removed the handle to that pump temporarily, and the outbreak stopped. The story goes that there was a family living in the building that was right up next to that, and people just didn't know better, but their latrine essentially was right next to this well, and so was going to be contaminating it. So you can imagine people were going to be sick regardless, but cholera was a particularly awful thing to get sick with. And that family had an infant that had traveled from some other place bringing an infection of cholera. Now, the infant's stomach was not fully developed, and so it was not quite as susceptible to the infection. So it actually continued with mild symptoms. Just the physiology of an infant is going to be a lot different. So it turns out that the infant was able to survive, I think, lived through the infection, but also meant that that infant was donating Vibrio cholera to the well for the better part of a week or two. So it was a longer-term source of a contamination in the well. And so ultimately that was theoretically the infection zero. The initial infection was an infant living in that spot. So kind of ties a lot of our water and wastewater treatment together here. So that's sort of the story of the ghost map. Very morbid, and also enlightening, and gives us a lot to be thankful about for modern plumbing and sewage water treatment. Okay, any questions about any of that? By the way, the one tip I have, if a lot of people here like to eat raw oysters. Any fans here? Okay, maybe not willing to admit it after all this right now. So there's some risk there. You probably already know, because they're effective filter feeders, and they can get stuff. Cholera can even be one of them. Of course, you'd have to get a lot. So if you have some sort of wastewater meeting the oyster beds or whatever, that's a potential danger. So if you decide to eat raw oysters, I recommend doing so on an empty stomach, because you have a lot more stomach acidity if you've not already filled yourself with other food. So if you're going to do something dangerous like that, again, if we added some cholera in here and I had to drink it, I'd make sure to drink it once I'm pretty hungry, right? And I'd probably do so slowly. So, yeah.
Isn't that also why you add hot sauce on it, because it helps with that?
So hot sauce, maybe so. I think a lot of hot sauces are probably pretty acidic. A lot of them have vinegar base. This is also why ceviche is a way to cook sort of shellfish and fish without actually cooking them, because your ceviche is taking typically like raw seafood of some various sorts, and sort of marinating it in lime or lemon juice, highly acidic conditions, and giving that some amount of time, right? So some of you look grossed out by this.
Yeah, my cousins did that one year for either Christmas or Thanksgiving.
Uh-huh.
Guess who got sick?
It was you.
Everyone.
Everybody. So there's probably a lot to concern yourself about, to do it well and to get a good clean source of stuff. So yeah, I don't know I would try it myself, but if I were to do something raw for myself, these are the types of things I'd be thinking about, because in theory there are ways that it can help if not work properly. But yeah, I think that's pretty brave to try it yourselves.
Oh, me, my mom, my dad, my brother, we didn't try it. Everyone else did.
Oh, okay.
They were just laughing at him. We told y'all.
Well, glad you were saved the ordeal there. I also, last time I had something that was probably some sort of food infection. I think I ate at this weird restaurant in North Carolina, and I think they had they were serving water that had been sitting, like steeping with some like mint leaves, and it was like, you know, whatever, maybe some lemon in it. But I was just like looking at it, I was like, how long has that just been sitting there, touching fresh produce that may or may not have been washed properly? And it's just like, you know, sitting out in, you know, in the sun or whatever. It's just like that. I don't really trust that water. But I drank some anyway, and I think I ended up regretting it. Okay, so how about our disinfection technologies? What are our options? What's typical? And kind of what are the thoughts on when and why to use them? Well, free chlorine is going to be the first and most common one we see, kind of the most obvious. And you actually know this as household bleach. Sodium hypochlorite is probably what you'll see in almost any household bleach stuff. And you know, a container might be something like anywhere between 1% and 5% sodium hypochlorite. Now, in the household setting, that'll typically have some base in it in order to push it towards the more stable, longer-lived, less reactive, less volatile form, which is OCl minus. So we can have chlorine gas. If we bubble chlorine gas through water, which does work, although chlorine gas is very dangerous and requires a lot of safety precautions. So in the water treatment field, this is a major potential hazard and something to take very seriously and carefully. But what happens if we do that, it very conveniently gives us, well, Cl minus, just chloride. Chloride is not reactive. It's happy to sit there with a minus. But it also gives us a chloride that has attached to the water, so ClOH, or HOCl as we often write it, plus one proton will come off of that water as well. I'm going to go ahead and rewrite this instead of ClOH. We see that it's kind of comboing with the water that way, but you'll see almost always we write it as HOCl, so just to be a little consistent there. So that's very commonly done. It's just chlorine gas. We add it to water. It reacts with water. It makes it more acidic, and it gives us hypochlorous acid. So HOCl is what we would call hypochlorous acid. Now, this hypochlorous acid will be in equilibrium. Let's see. I guess I gave myself space to write this up here. So hypochlorous acid, HOCl, will be in equilibrium with its conjugate, OCl minus, so it can just pop that proton off, plus that proton. So as soon as we add free chlorine into our water, it doesn't really matter if we add it as this chlorine gas, the HOCl, we could add NaOCl, and you know what I can do, I've already written this up once, so I apologize if I'm making the notes convoluted for you, but I think I'm just going to use this space a little more cleanly this way. If we, let's say, add NaOCl, that would give us a sodium and an OCl minus. So regardless of if we're adding it as HOCl or as OCl minus, those two are going to become equilibrated based on the pH. So the pH will dictate how much HOCl versus OCl minus we have, and if we have one, then we know we have the other, just in some ratio, right? That's the thing about equilibrium. So anytime we say we have free chlorine, this is really what we're talking about. We have those two things, and the ratio depends on the pH. Okay? It turns out HOCl is a much better disinfectant. So HOCl is between 5 and 10 times stronger, faster rate constant, higher rate constant for disinfection for almost every organism. So it is more reactive and will disinfect things better. And so that's why we actually care a lot about our pH during disinfection. So we'll adjust it to make sure we have as much HOCl as we think we have, or we design it to have. Okay, so then there's combined chlorine. I mentioned this. That's when we take some sort of chlorine of some kind, free chlorine, OCl minus, HOCl, whatever. We combine it with an amine group, and then we have an ammonia, so NH3, except that now it's maybe NH2Cl. So instead of one of those hydrogens, now we have one chlorine. This would be monochloramine. We could also have NHCl2, which would be dichloramine, or NCl3, which is basically just we've stripped off all of the protons and now we have trichloramine. Okay, this is the reason you should never, ever, ever combine bleach and ammonia when you are doing household cleaning stuff, is because these chloramines are more volatile. We do use them on purpose in small amounts, but if you were to wash your toilet and have a bunch of bleach in there to bleach stuff, and then you figure, oh, it's really dirty, let's add some ammonia too. Suddenly you're making a lot of this stuff, and some of it's going to come off in the gas phase, and you're going to chlorine gas yourself, chloramine. Some reactions like that you might even get Cl2 gas. I don't think so. I think that's mostly chloramines, but you want to be careful because these are no fun. Chloramines are also pretty common. You've probably experienced them a lot if you've ever gone swimming. Have you ever felt like some pools just have a lot of chlorine in them? You can smell it, you can feel it, burns your eyes a little bit. That's actually a sign of lots of chloramine. That's not necessarily the free chlorine. It'll have a little bit of a smell for free chlorine, but not nearly that. Oh, there's so much chlorine here. And free chlorine actually doesn't sting your eyes very much. It's the chloramines. Now, how do they get there? Well, chances are that, amines of some sort were added, which usually means uric acid, which usually means someone peed in the pool or has sweated a lot in the pool. It's kind of the same. So if you find a pool that smells very much like chlorine, well, yes, it's got the chlorine, that's good. Unfortunately, it's got some sort of source of ammonia in it too. Okay. So another option, so chloramines, by the way, this combined chlorine, we would not use that as a direct disinfectant. That would be a sort of a secondary or residual, typically. So we will do that. We will often try to have some free chlorine residual in our water treatment lines, but we'll also typically have a little bit of it as chloramine to just make sure that it's long lasting there. All right, chlorine dioxide, ClO2. This is another form of chlorine. It would act a lot like free chlorine, but the chemistry is going to be a bit different. We're not going to have that equilibrium situation. It's not as commonly used. But its use would be pretty similar to free chlorine. Okay, then we've got ozone. Heard a lot about ozone. variety of different scenarios, right? We like it up in the upper atmosphere where it's blocking all the UV from coming to us and, you know, protecting us. We don't like it so much here where we're breathing because it's an irritant. Literally, it's an oxidant, right? So it's causing oxidation reactions in our lungs and would bother your asthma or whatever. It's just a natural oxidant. So there's going to be a little bit of it. And if we apply it to water, we can generate it in several different ways. If we apply it to water, then we can use it as a disinfectant. It's pretty highly oxidizing. We can also sometimes combine this with UV light or some other things to cause it to split apart and make radicals. That would be an advanced oxidation process, like we mentioned below. So we'll come back to that. We could also use UV light just by itself directly in the right spectrum. This is something that I've focused a lot on myself with my research, so we'll talk a fair bit more about that. But that's definitely an option. And that one we will, as we'll see, can cause DNA damage directly and prevent organisms from having the capacity to reproduce. So advanced oxidation processes are actually not typically used for disinfection exactly. Usually we're using that as a way to destroy some other chemicals. Maybe we're worried about pesticides in a lake that receives a lot of water from different, you know, farmland or golf courses or whatever. You know, there might be some chemicals in there, and we want to make sure to try to destroy as much of those as possible. We're going to add some step that's pretty harsh. We could do like super fancy filtration, but sometimes it's more cost effective to use an advanced oxidation technique. Certainly this would also destroy lots of viruses and bacteria. So you might be able to do just this and then chlorinate your water for your residual. But typically these are designed with not necessarily disinfection in mind, but it would likely do that. Okay, so let's talk about chlorination. I think next time we'll talk about UV light. In I think 2 or 3 weeks, I'm going to have to, I think it's Thursday, maybe the Thursday that's near the end of October, I'm going to have to miss a class. So I might have one of my students come in and give a guest lecture. I'll get back to you on that. So I might have a special topic on like advanced chlorine as like an advanced oxidation process. But for now, we're going to talk about chlorine. Today, I think we'll get to UV next time after the break. So I already mentioned this, but HOCl is the stronger for free chlorine. That's the combination of HOCl and OCl minus. HOCl is the stronger one. So we typically want slightly acidic conditions, and that's because we have some equilibrium constant, right? You've seen this equilibrium constants where this is going to be equal to the products divided by the reactants. That's how we're defining our equilibrium status, right? And you'll recognize the P. If we say pH, that's the negative log of something. So we happen to know that the Ka for chlorine is usually, this will be temperature dependent a little bit, but it's going to be pretty close to 10 to the negative 7.54. And if we talk about it with a pKa, that means the pKa is 7.54. That means when the pH is at pKa, these guys are the same. So let's write that out. Let's say if Ka equals, let's just say that we have H plus and then the other two things are equal. When equal, HOCl and OCl minus. Now this does not, what I'm saying here is they're actual equal amounts. We can be at equilibrium with different amounts. if we have a really low pH, really high pH, we'll have an equilibrium. But right now, what I'm not saying equilibrium. I'm just saying like when these two have the same amount, then what happens is we'll just say, let's just say each of these equal to 2, for example. So this would be that times 2 divided by 2. These will cancel. And so we see that Ka is equal to the H plus concentration when those two are equal, right? That's the equal point. And so we could say that pKa equals pH at this condition, right? So that means the pH at which the OCl minus and the HOCl are equal should be 7.54. Okay, and I'm sorry, I should have gone ahead and written this up here. So what we're talking about is this equilibrium between HOCl that can dissociate into H plus and OCl minus, or it can go backwards and reassociate, I guess, do the reverse reaction to HOCl. That's just going to happen. That'll be pH dependent. When the pH is equal to the Ka, or the pKa rather, we get the same amount. So if we're operating at neutral conditions, like pH 7, this is slightly favoring HOCl. Anything above 7.5 will be favoring OCl minus. So we generally want slightly acidic. That's that 5 to 6 range. And we could calculate with this equilibrium reaction how much of percent of one or the other would we have if our pH was 5, for example. So let's do a problem on that. And actually, before we do that, I think I have a slide to kind of just show this graphically. And I want to connect that if I do. No, I guess I don't. Let me just draw a little bit real quick. We've probably looked at this before, and I know we will see more of these. But if we draw a graph of pH versus what we'll say is the fraction. So if 100% is one form or the other, this will be at 1. And so we could say this is pH 7. So we'll note those little 7.54. Call this 8, 9, 10, and so on. So 6, 5, 4, 3, 2, 1. Not quite drawn to scale, but you get the picture. So let's just kind of focus in that area. So at first, we have almost all HOCl. So about 100% will be HOCl, and it'll stay up pretty high up until we're getting close to that pH 7.5 mark, and I did not draw that very well. I'm going to draw it through the halfway point. If this is 0.5, like half of it should occur right about there. So I'm going to try to draw this a little better and say it should look something like that. On the flip side, when we draw our HOCl, excuse me, our OCl minus, that will also be almost 100% as we're up near pH 14. And then it'll do the same exact thing in reverse. Cross through there. And we'll pretend I drew a nice smooth transition there. And it would look like that. So you've seen these plots before, I'm sure. You'll see them again. We'll do a fair amount of these with this water softening kind of stuff. This is the equilibrium chemistry, and this is what I mean when I say we've reached that equal point. We can have equilibrium at any one of these places. That doesn't mean there's equal amounts. But the point at which we have equilibrium and equal amounts is at 7.54. Okay, so let's do a problem. If we have 15 milligrams per liter of HOCl that's added to water, and we're not really given any other information, so we have to assume it's 15 milligrams per liter as HOCl. for disinfection and the final measured pH is 7, what percent of HOCl is not dissociated? It says assume the temperature is 25 Celsius. That's so that we can look up some Ka value, and I'm just going to say we're going to use the 10 to the negative 7.54. So really this is an equilibrium problem, and this will give us some sense of what's typical if we were to disinfect at a neutral pH. So give that a couple minutes, make sure you're feeling good about that. I'll walk through it with you. All right, so there's a couple ways you can go about this. One is, really you'll end at the same place. One is the long way. And that is if you fall for the trap that they laid for you here in the problem. They said 15 milligrams per liter is added, and then it goes on. And it probably puts your mind into thinking, okay, we've got to think about this in terms of what mass is here, we'll convert that, and find the actual, we could solve it for, okay, what is OCl minus? We get some number. What is the HOCl? Get some number. And then do a fraction analysis, right? What is this one divided by these two, which is kind of what I wrote here, right? There's the HOCl divided by HOCl plus OCl minus. That's fine, but what you might notice is that you don't actually need the numbers, you just need the ratio between them. So you actually don't have to use this 15 milligrams per liter, because you'd take this and say, multiply that by 100%, and that gives you your answer, right? So you actually don't have to even get into the mole conversions or any of that, because you're just asked about what percent is there. It just is going to take a little bit of algebra to make use of this equation, the Ka equation, in order to solve this in terms of pH. So the easiest way to do that is just to simplify this guy as much as you can, and you'll notice there's an HOCl on the top and the bottom. So if we divide the top and the bottom by HOCl, we'll get 1 over 1 plus OCl minus divided by HOCl. So the good thing here is now we have it in terms of a ratio. Our what our final answer is going to look like, is in the form of a ratio between OCl minus and HOCl. And that's good because Ka defines a ratio between those two. So anytime we're asking for some kind of a fraction or a proportion, that inherently you can sense that that's a ratio of some sort. So we should be able to adapt this into a direct ratio. That's what we've done. So now we can take a look at our equation over here and say, well, can we do anything to this guy in order to fit it in the same ratio to substitute into here? And you don't have to look too hard to find that if you divide both sides by H plus, you'll have that OCl minus to HOCl ratio. And worst case, you could just flip it, but you'll see that we could get this if we divide Ka divided by H plus. That's just coming from that equation. So then, now that we have that, we'll substitute that into here and be able to solve our answer, right? So then we say 1 over 1 plus Ka over H plus. So now the only variable here is H plus, which was given, and Ka is known. We're going to multiply that by 100%, and we can get our answer. I haven't done that yet, so if you guys have it, I would assume that it's going to be somewhere just a little bit above 50%, maybe like 60 or so. Anybody have that?
78.
Okay, that sounds correct to me. And we'll I'll toss it in Excel just to have an added confirmation. But you said 78 point what?
It was 77.6.
77.6. Okay. All right. Anybody else confirm that, or? I think that should be just right. So let me. Okay. Great. So that's how you would find the HOCl, and you can see we can do that for any pH. And then if we needed some designed amount, let's say we needed exactly 5 milligrams per liter of free of HOCl for our disinfection process, we could then be thinking, okay, well, at what pH do we have to, is our limit, right? At pH 14, we have 0.000001% of the solution is HOCl. So we're going to have almost none of it. So we could find a particular pH that we could reach some minimum. So we're going to leave it off there. We'll talk about some disinfection byproducts, things like that next time. But have a good break. We'll see you in a week.